So without further ado, let's get started with Flowing Waters, exploring our freshwater ecosystems with Eva Anderson, Laura Croll, Lucca Sterrer, and Megan Locher. Come on up. Who's going first? Yes. Right here, and then there's the one. Does it work? Yes. You want to pass it really quick? Perfect. Okay. Yeah. Okay.
Hi, everyone. We called our presentation Flowing Waters, Exploring Our Freshwater Ecosystems, because we all chose to study or are still currently studying freshwater ecosystems in some capacity. My name's Eva Anderson. I'm a second-year employee student, and I'm going to be presenting on my ecology thesis looking at macroinvertebrates in the fens.
My name is Laura Croll, and I'm going to be presenting on my thesis about assessing beaver dam antelope-based restoration efforts by evaluating aquatic macroinvertebrate communities.
I'm Lucca Sterrer. I'm a second-year MSE candidate, and I'm presenting on my thesis research for the harmful algal blooms on South.
For those of you who weren't here 30 seconds ago, my name is Megan Locher. I'll be presenting on my MEM project today, looking into the impacts of wetland restoration efforts.
So before we dive into our individual projects, we kind of wanted to set the tone with a fellow women of STEM, Rachel Carson. She's a pioneer of environmental awareness. She once said, "In an age when man has forgotten his origins and is blind even to his most essential needs for survival, water, along with other resources, has become the victim of his indifference." And we just wanted to set the tone here because we want to remind everyone as we talk about our projects with freshwater why it's important for all organisms on life.
So to start off, freshwater ecosystems are greatly threatened by climate change. And the 1.5 degrees Celsius warming per year has altered Southwest hydrological regimes. We're witnessing a freshwater biodiversity crisis. And the freshwater biodiversity has decreased by 83% since the 1970s, which is very substantial.
And we wanted to highlight the importance of wetlands again in this, in saying that recently in 2022, there's been research indicating that there's preventative costs of up to $745 per acre when it comes to flooding in the southeast of the United States. And so our projects are centered around the health and preservation of wetland or freshwater ecosystems with a focus on those within Colorado.
So I'm going to be presenting my thesis work first. It is a macroinvertebrate community composition assessment within fens of the San Juans, pre and post mining pollution restoration efforts. So as we were just discussing how important freshwater ecosystems are, I'm going to be talking about one in specifically, and these are wetlands. They are also incredibly important, but possibly up to 70% of them are already destroyed. Wetlands in mountainous regions are also very vulnerable to climate change as they depend heavily on the hydrological or climate-related factors, including precipitation, evaporation, and snowmelt. Specifically, wetlands in Colorado cover 1.5% of the state, but up to as much as 90% of Colorado's fish and wildlife depend upon them at some point in their life cycle, making them incredibly important. A specific wetland I am going to be discussing is fens. Fens are peat-forming wetlands that rely on groundwater input. Peat is organic matter that forms in waterlogged conditions where organic matter accumulates faster than decomposition is occurring. Peat, they take thousands of years to form, and they're very vulnerable to disturbances and are also very difficult to restore once impaired. In southern Colorado, it takes about 2,000 years to accumulate 8 inches of peat, suggesting that these fens are between 2,000 and 10,000 years old, and they're typically found between 9,000 and 12,000 feet. Fens provide many ecosystem services. These include acting as carbon sinks, filtering pollutants from water, supporting very high biodiversity, as well as rare species. However, as many wetlands have been, they've been greatly impacted by human activities. This includes mining, reservoir construction, irrigation for agriculture, water diversion, and livestock grazing. And all of these can reverse that 10,000 plus year process of peat accumulation. Climate change, as I mentioned, is also a threat to fens in particular because it can affect how much groundwater is reaching them, which could lead them to dry out. As I mentioned, they support very high biodiversity, and this is for a couple reasons. So they are fed primarily by groundwater, and this leads to the waters within being more basic and very high in minerals, allowing them to support very high biodiversity as well as species that are disjunct from their native ranges. So my research objective is to compare macroinvertebrate community composition and taxonomic richness before and after mining restoration. So my study site is in the Bonita Peak Mining District in the San Juan Mountains. This area has metal deposits of lead, zinc, silver, and gold and has been historically mined. Mining activity began in the 1860s and mostly ended in the 1920s, and this was designated a Superfund site by the EPA in 2016. And this is just a zoomed up image of my site on a map as indicated by the red arrow, and you can just see how many mines were in that area. Just a lot of mining activity. So we're particularly interested in 2 sites within the San Juan Mountains. And the first is the Burrows Creek Fen Complex, which is indicated by image B, and this is acting as my degraded site. This is located north of Houghton Mountain. It falls at 11,860 feet of elevation. Houghton Mountain has naturally high metal loading into its streams and soils, and in addition to the altered seeps and groundwater runoff from past mining, which has contributed to increased loading on the site, leaving us with a very degraded fen complex. In addition to impacts from mining activity, a trans-basin water diversion ditch was created for private water use that was diverting 11 cubic feet of water per second. And this has since been diverted back, but this did heavily alter the watershed and influence erosion. My second site is the Pirate Ship Fen Complex, indicated by image A, and this is my reference site. It was restored approximately 11 years ago, and it falls at 11,739 feet of elevation. So I am partnering with the Mountain Studies Institute for my thesis, and they did carry out restoration on the degraded Burrows Creek Fen Complex this past summer. And they did that with 3 overarching goals. And the first was erosion control. So erosion on the side of Houghton Mountain has resulted in sediment deposition into the fen, and this has been identified as the main cause of disturbance. So to prevent future burial of the fen, check dams, media lunas, and one rock dams were installed down Mount Houghton along the 3 main gullies leading to the fen complex. And then the erosion channels were also revegetated. The second goal was to restore the hydrology. So ideally the area is being re-wetted over this past year, and they did that by removing excess sediment that was deposited into the fen complex using an excavator. And rock check dams, media lunas, and one rock dams were placed perpendicular to the roads along Mount Houghton that were originally diverting water away from the fen complex to re-divert that water to go back down to the fen system. And the last goal was to restore native vegetation. So native vegetation was replanted, and this includes sedges, sphagnum moss, grass, and shrubs. Lastly, drone surveys are being used to monitor erosion on the hill slope, vegetation cover survival, and the re-wetting of the fen. So I went out in June 2024 for my first round of sampling. We sampled a total of 22 ponds. 6 were mine adjacent, 9 were lower burrows, and 7 were in the reference site. And before any macroinvertebrate sampling occurred, we measured our environmental factors, which included pH, dissolved oxygen, temperature, and conductivity. Then we did sample the macroinvertebrates, and we used a D-net sweep where we swept across 1 meter of substrate for 10 seconds in each fen pond. Macroinvertebrates were then placed in ethanol after large debris was removed. After all the sampling was done, came all the lab processing, which included elucidating, sorting, and identifying all the macroinvertebrates to genus and family. Here's just some photos. This is a mine adjacent fen, and you can just see it doesn't look healthy compared to a fen that's further down away from the mining activity. These are the bins that we sort of looked at the macroinvertebrates we were seeing. These are my advisors, Dr. Houston and Dr. Washko. This is a DNet that we use, and here's just another really beautiful picture of the site. These are some of the macroinvertebrates that I got to see in the lab. These include dragonfly larvae, some copepods, some water boatmen, some diving beetles, fingernail clams, caddisfly larvae, and a lot of dipteran larvae and pupae. So I did wrap up year 1 data analysis, and that's what I'm going to go through now. So first, we assessed the environmental conditions by treatment to determine if there were significant differences. And a Kruskal-Wallis test showed that pH and conductivity varied significantly by treatment. pH was lowest in the mine-adjacent ponds, and conductivity was highest in the mine-adjacent ponds. We also looked at richness, which is the number of taxa found within each fen, and an ANOVA showed that richness differed significantly by treatment. Taxa richness was highest in reference ponds and lowest in mine adjacent ponds. One second. Additionally, all taxa were assigned a tolerance value using a Hilsenhoff's family level biotic index, where index values fall on a scale from 0 to 1 and can be applied to water quality. 0 would represent zero would represent excellent water conditions, and 10 would represent very poor water conditions. A Kruskal-Wallis showed that the average tolerance values differed significantly by treatment. But interestingly here, you can see that the average tolerance value was lowest in my mine adjacent ponds, which suggests that these support more sensitive species. However, this is likely because of the very low invertebrate density that we got in those samples, which didn't necessarily allow for a super accurate Hilsenhoff family level biotic index to be calculated. And lastly, I made a non-metric multidimensional scaling visualization to compare community composition of the sites. So this image is showing ordination plots of the 2024 data, where each point represents the taxonomic composition of a fen, and the closer 2 points are, the more similar their community composition, and vice versa. So a Permanova did show that differences in community composition were significantly different. You can see the mine adjacent sites clustering further to the right of the graph compared to the lower burrows in the reference sites clustering to the right. Additionally, you can see that lower burrows and the reference sites overlap, indicating similar community compositions, but you can see that the reference site has a much more constrained community composition compared to lower burrows. Environmental vectors were also mapped onto the NMDS graph and show pH moving into the lower burrows and reference sites, indicating higher pH in those systems, and conductivity moving into the mine adjacent, indicating higher conductivity in those fen ponds. Lastly, I just want to quickly touch on the broader significance of this. There are very few studies that have sampled macroinvertebrates in fens of the Rocky Mountains. And this is the first study to track macroinvertebrates in fens pre and post mining pollution mitigation, and it will provide really critical information for future fen restoration. I want to thank my advisors, Dr. Susan Washko, as well as Dr. Derek Houston, as well as Scott Roberts with the Mountain Studies Institute. He's been incredibly helpful. And my next steps are just to go out sampling in next month, actually, and repeat the process so that I have the 2024 data that I'm going to compare to the 2025 data. Thank you.
All right, thanks, Eva. So now we have time for questions. There are QR codes on the back of the seats. If you'd like to scan those, you can enter your questions for Eva there. Or folks on Zoom, you can put questions into the chat and they will find their way to us. While we wait for questions to pop in, Eva, from your perspective, what's the difference between the mine adjacent and the lower fen ponds? They're in the same drainage and the reference is outside, yes?
Yes. Yeah, so that might be a little confusing. We are only sampling 2 fen complexes, but the location of the ponds is a little different. The mine adjacent ponds are exactly adjacent to all the mining activity, whereas the lower burrows were quite a bit down the drainage. And as those photos I showed earlier, you could see just how different they looked. So yeah.
Awesome. Someone from the crowd has this question for you. What did you find to be the most interesting aspect of your research?
I really just love looking at macroinvertebrates under the microscope, and I just think they're so fun to look at, and everything is so beautiful. And I think that it was really fun to find the fingernail clams in there. They're just, yeah.
What techniques are used for general restoration or fen restoration? What does this preserve fens functioning as carbon sinks?
That's a good question. I would probably have to do more research into that. I know that replanting the native vegetation is really important for supporting the species that typically would go to those fens, as well as just preventing that fen from drying out. As I mentioned, as soon as that fen dries out, that peat is going to dry out, and all those ecosystem services will no longer occur. So I think the main thing is obviously removing any sediment that might be deposited on the fen and revegetating the area, and keeping foot traffic, recreational traffic away from the fens.
All right, we have time for one more. Why do you believe so few macroinvertebrate studies have been done with remediation in fens?
Yeah, that's a good question. I think yeah. I think fen sometimes can be hard to access, and a lot of fen research is on the vegetation, and less so on the macroinvertebrates, even though they are really important indicator species. That's a really good question. I'm not totally sure.
All right, thank you so much, Eva. A round of applause. All right, next up we have Laura Croll.
So as already introduced, my thesis is on assessing beaver dam analog-based restoration efforts by evaluating macroinvertebrate community composition. So a similar idea as Eva, but with BDAs. I'm going to start off by talking about mountain stream degradation in the Rocky Mountains. Small streams are fundamental ecological and recreational facets of Rocky Mountain landscapes. They make up more than 2/3 of the total extent of stream habitats, stream networks. They're of critical importance in biotic diversity, hydrologic connectivity, and the downstream delivery of nutrients. However, land use history has severely impacted headwater streams. Livestock grazing, mining, and logging have degraded streams and mountain habitats. As a result, the geomorphic complexity in mountain streams has been reduced. A significant issue arising from reductions in stream complexity is disconnection from the floodplain. One factor contributing to lack of floodplain connections is a reduction in beaver activity compared to historical levels. Beaver trapping and extirpation has led to lowered shallow groundwater tables, cutting of river channels, and ultimately a transition from complex wetland systems with embedded floodplains and braided channels, as we can see on the left in the beaver maintained habitat, to singular channels, as we can see on the other picture. So beaver dam analogs, short BDAs, are a novel and highly promising stream restoration strategy. that is increasingly being implemented across much of the historic range of beavers in North America. BDAs are channel-spanning structures that are strategically placed in streams to mimic or reinforce natural beaver dams. They present a very attractive alternative for habitat restoration at locations with insufficient resources for supporting beaver or where beavers would be viewed as a nuisance. Here we can see what a BDA looks like. A goal within BDA installation is to restore degraded fluvial ecosystems, thereby improving in-stream habitat, riparian habitat, and species heterogeneity, as well as reducing stream channel incision, raising groundwater levels, and storing water. Just like natural beaver dams, BDAs change the flow of water and sediment, even if temporarily. Because this method has only recently been implemented in some areas, data are still lacking on how streams respond to BDA-based restoration efforts. The lack of monitoring after BDA installation with regards to aquatic invertebrates and overall biological responses leaves questions about whether the restoration goals have been met. Recently, there has been a call for additional investigation to evaluate long-term responses to BDA-based restoration. This can inform future land management practices. Previous work describes how beaver-induced alterations to stream morphology significantly change aquatic macroinvertebrate communities, indicating that BDAs also affect aquatic communities. Next, I'm going to talk about aquatic macroinvertebrate functional traits. Different macroinvertebrate taxa have specific habitat requirements, so changes in macroinvertebrate communities can indicate alterations in stream physical and chemical characteristics. A suite of insect functional traits, such as feeding habits and mobility types, are expected to shift in response to changes in habitat. Aquatic macroinvertebrates are categorized into functional feeding groups, short FFGs, according to their modes of feeding. These include filterers, shredders, gatherers, grazers, and various predators. Mobility groups, on the other hand, classify macroinvertebrates based on their feeding habits, movement habits. These include burrowers, clingers, swimmers, sprawlers, skaters, and climbers. Because the traits of the invertebrates will match the habitat available, noting which traits are present in an area can provide insights into how macroinvertebrates are responding to habitat changes in streams. So what were we expecting to find in my project? In the untreated section, we're expecting to see invertebrates with flat bodies adapted to live in faceted water. Those FFGs would be grazers, gatherers, and filterers. And for MGs, we were expecting mainly clingers. These groups would be stoneflies, clinging Coleoptera, clinging mayflies, free-living caddisflies, and black fly larvae. And in the BDA treated section, we were expecting more lentic taxa with the dominant FFGs of predators and shredders. And for MGs, we were expecting mainly swimmers, burrowers, and sprawlers. These are dragonfly larvae, large case building caddisflies, swimming mayflies, swimming Coleoptera and Hemiptera, Diptera larvae, and multipods. Next, introduce the study site, which is located at Trail Creek in the Taylor River Basin, where several BDAs have been installed for wetland restoration. While the BDAs resulted in widening the reach of inundation, the progress and impact on these measures on biota have not been monitored. Like many streams in the Gunnison Basin headwaters, Trail Creek became a simplified, incised stream due to anthropogenic influences and the absence of beaver. The goals of the BDA installation at Trail Creek are to reinstate the wetland conditions that support natural processes supported by beaver activity. This includes substantial increases in pond area, as well as higher water levels. So as we can see from these before and after pictures at Trail Creek, the BDA treatments have led to changes in inundation. However, changes in fauna Specifically, aquatic macroinvertebrates have not been evaluated. So the goal of my project was to compare aquatic invertebrate communities in the restored section of Trail Creek to the upstream unrestored section, as well as a nearby section with natural beaver dams. The composition of these macroinvertebrate communities, combined with the functional groups and traits, can provide valuable insight into the stage of wetland restoration. So here is the map of my study area, where we can see furthest downstream is the BDA section, where those BDAs have been installed. A little further upstream, we have an untreated section where there was no changes to the stream. And furthest upstream, we had a natural beaver dam section. I'm going to briefly talk about the methods. So the invertebrates were sampled from each side using a timed D-sweep. And then we also assessed additional habitat variables, which were water depth, water velocity, and substrate grain size. Statistical analysis was conducted in R, and we ran statistical tests to compare the makeup of invertebrate communities on the different sites. And we used traditional community ecology statistics to accomplish this, which were these tests. And now on to the exciting part, the results. So 49 different families were identified across all 30 samples that I took, so 10 samples from each of the 3 reaches. And those mainly belong to the families of Diptera, Trichoptera, Ephemeroptera, Plecoptera, and Mollusca. And in total, I processed 6,488 invertebrates, which was really fun. And I ran a lot of tests. There's a lot of graphs, and for sake of time, I'm going to skip over them and talk about the nitty-gritty of the results. We also ran an indicator taxa analysis with indicator taxa for each reach, which I also unfortunately had to skip due to time. And now on to the summary of the results. So we saw that the BDA treatments meaningfully altered physical habitat. They led to slower flow, higher depth, and smaller grain size. We also saw distinct shifts in functional and trait-based assemblages. We saw at the BDA section, we saw the main FFGs of gatherers. For MGs, it was burrowers. For substrate associations, it was taxa that were both erosional and depositional. And for habitat, it was taxa that were both lentic and lotic. So in summary, those are all taxa that are adapted to deeper waters, smaller grain size, and lower velocity habitats. Community composition differences were based more on the dispersion than on taxa present. So this means that the BDA reach seems to be more homogeneous than the other treatments, so there's less variability. The environmental variables were not correlated with reach-wide community compositions. This leads us to think that other drivers might be at play. So possibly more time, how cemented is the substrate to the ground, biological interactions, and legacy effects, i.e. past beaver presence. So biological responses to BDAs may be mediated by species-specific dispersal ability or colonization history, rather than immediate habitat conditions. Environmental changes influenced which invertebrate traits were present rather than how many different families. So the same taxa were present in a lot of the samples, but in slightly different proportions. Now onto the discussion of these results. So the important question was, did the BDAs create wetland conditions at Trail Creek? We had to say no. However, well, this was because obligately lentic taxa that are characteristic of nearby wetlands were absent, which would be dragonflies, swimming beetles, and possibly other Diptera. We also did not find enough difference between treatments to classify the BDA reach as a wetland. So why was the goal not accomplished? Possibly more time is needed. Then also there are BDA construction choices that need to be considered, and they need to go with the specific habitat that the BDAs are implemented, which include the dimensions of the BDAs. They need to be fully channel spanning to allow for more ponding and create wetland. And then they were also leaky. There was water flowing through the leaks, further decreasing the ponding effect and letting the water flow too fast. And then finally, spacing needs to be considered. So how far are the BDAs spaced apart? So the simplest and most important take-home message of this project is that future monitoring, especially over longer timescales, is key. And also a lot of specific considerations need to be made when installing BDAs for wetland restoration. Thank you.
All right, thank you so much, Laura. Just a reminder, the QR codes are on the back of your seats if you want to submit some questions. We have time for a few. One person asks, what is the next step in your research?
I think, well, my project is done, but I think this is a very interesting site and a very interesting project that would, as mentioned, really benefit from future monitoring. So I think it would be great if there would be more research at the site, maybe assessing the BDAs and seeing what can be improved on the structures. And yeah, further monitoring every year would be great to see how the community changes from year to year.
Thanks, Laura. Any other questions from the audience? All right, I'll ask one more. What was your favorite invertebrate that you found during your research?
Heptogenia, aka I called them pancake face. They're mayflies that are really flat and they can like cling to stones. And so they're adapted to fast flowing water and they just look really cool.
We love a pancake face. Are there other studies that you found looking at how long it might take to see different macroinvertebrates moving in?
So based on my research and my reading of studies, it really depends on the family and on the habitat. So some of them are quicker to come to a new habitat, and for others it might take longer. But usually invertebrates are expected to be really quick in finding an appropriate habitat if it's present.
Do you see yourself working with macroinvertebrates in your future?
Yes, I have to say I'm a bug person now. I really like bugs. They're really cool. So yeah.
Awesome. Well, we expect to see some upcoming theater productions surrounding
Yes.
macroinvertebrates. With that, let's give Laura a round of applause. Thanks so much. All right, and next up we have Lucca Sterrer. Yes.
Okay, so we're gonna deviate a tiny bit from the macroinvertebrates. I did my thesis research on the harmful algal blooms of Blue Mesa Reservoir, and we did a bit of a trophic case study looking at different organisms in the reservoir. So before I get started, I just wanted to do a general thank you of some of the organizations that have helped out with this project. We had a bunch of fieldwork that had to get done last year, and it could not have been possible without WCU, the Park Service, CPW, and the US Geological Survey. And then EcoAnalysts is a lab in Idaho that we sent our phytoplankton out to. And then our funding source, of course, the Upper Gunnison River Water Conservancy District. And then just really briefly, some of the specific people that have helped out a bunch in this project. My two wonderful committee members from Western, Derek and Hannah. I also had a field partner, Bridget, who helped out with the majority of the field sampling, and our boss, Nikki, who helped out with the planning. Several CPW members, including Bill Pate, Adam Hansen, and Dakota Pincus, helped us out with the majority of the kokanee salmon research that I'll get into. And then I also had a pretty awesome undergraduate lab technician who has been helping me out with the majority of the phytoplankton identification. So I've been with the Park Service at Keir County since 2021, and since 2018, the Park Service has been closely monitoring the reservoir for these blooms. So algal blooms are a rapid overgrowth of algae that can produce toxins dangerous to both humans and wildlife. And they're becoming more frequent across the West due to warmer temperatures and increased nutrient runoff directly from agriculture. So we're seeing that Blue Mesa hasn't been immune to these phenomenons. So my project takes a look at the history of the algal blooms on Blue Mesa, as well as a current overview of the ecosystem and the reservoir itself and how the algal blooms are affecting that delicate balance. So these two pictures on the right are ones that I've taken with my time at the Park Service. They're blooms that you can see on Blue Mesa. And before I dive into the logistics, I wanted to give a brief context of the organisms at the center of my project. So cyanobacteria are actually some of the oldest life forms on Earth, and their presence in freshwater ecosystems is completely natural. In small, balanced amounts, they're part of a healthy aquatic ecosystem. So the problem really isn't that they exist, it's how often and how intensely they're blooming now. So these HABs, as I said before, are being fueled by two major factors, nutrient-rich runoff and warming temperatures linked to climate change. And this shift has pretty serious implications. Algal blooms can reduce oxygen level in the water, releasing toxins and throwing off the balance of the whole ecosystem. So my project kind of builds on that existing research body by looking specifically at how these blooms are affecting Blue Mesa's ecosystem. So to really understand the impact of these algal blooms, it's important to look at the ecosystem as a whole. So we have phytoplankton, which includes the cyanobacteria as the bottom of the food chain. They're the primary producers. So when their populations explode during blooms, it can change the availability of food and oxygen in the water. And then the zooplankton, which feed on that phytoplankton, can either thrive or struggle depending on the type of algae present. So these effects move up the chain to the fish species, which rely on the stable food web and healthy water quality. So this is our process. We had 3 different sites in Blue Mesa. Blue Mesa has 3 different reservoirs for all of you that aren't familiar. So there's Iola and Cebolla and Sabanero. And we had a point in each basin that we visited 12 different days over 2024. We did a bi-monthly sampling process from about May to November. And the whole idea was at each sampling occurrence, at each point, we were trying to get a representation of the water column. So we did a stratified sampling method of collecting water from the 0 to 5 strata, the 5 to 10 strata, the 10 to 20 strata, and the 20 to 30 strata. And that was kind of to capture all of the whole water column. So our field research objectives, at each sampling occurrence, at each point, we were collecting stratified zooplankton toes to assess that community structure. We also did a Van Dorn sample to assess phytoplankton community structure, microcystin content, and chlorophyll A content. That Van Dorn sampler is kind of a giant tube that you lower into the water column, and it traps water from that specific spot. So we were able to kind of get a stratified basis of the community structure. And then we also did a full water column profile. So meter by meter, we had a multi-parameter sonde to collect dissolved oxygen, temperature, pH, and specific inductance. And then CPW also helped us out with the kokanee salmon abundance. They set vertical gill nets overnight and they retrieved them 48 hours later to calculate kokanee density. And when we got into the lab, the first thing we did was calculate chlorophyll A concentrations. And we utilized absorbance values from a spectrophotometer, which is essentially measuring how much light it's emitting. And then so Bill Pate with CPW has been processing zooplankton samples for them for like 10 years. So he was kind enough to process all 140 of our samples to determine species abundance and community structure. And then we sent half of our phytoplankton samples off to a lab in Idaho who did a lot of cyanobacterial abundance for us. And we're doing the other half in-house, so looking at identification, abundance, and community structure. And then we're also performing a ELISA test to check for microcystins. So an ELISA test is an enzyme-linked immunosorbent assay, which is a fancy term for determining how much of that toxin content is being emitted from the cyanobacteria. And then CBW also analyzed their gill netting data to determine the location and abundance of planktivorous fish. So I'm going to share briefly our preliminary results. We are still working on the phytoplankton identification, so I don't have those results for you, and we haven't started the microcystin testing either. But I have some water quality parameters, chlorophyll, zooplankton, and vertical gill netting data. So this first graph is a temperature plot for the reservoir. You can see that in Iola throughout most of the season, it's the shallowest basin. So it kind of stays isothermic throughout the whole season because it's so shallow that the light can penetrate all the way to the bottom. And then in Saboya and Savonniere, you can see that more stratified profile, just because of their deeper depths and the distribution of the water. And then these two graphs, I have a dissolved oxygen graph and a pH graph. Our pH probe had some difficulties this summer, so some of those values are going to be way out of the norm. But the dissolved oxygen graph is very telling. So as algal species die, they kind of lower in the water column and they lower the dissolved oxygen. So we see that impact kind of mainly in Saboya. That basin, for whatever reason, has really low dissolved oxygen. And that's a whole other study done by CBW that I am referencing in my thesis. And then these are some trophic status index plots. So this is the outcome of our chlorophyll data. We used those values from the spectrophotometer to plug into this trophic status index that would give us an idea of what the reservoir was doing at what time. So throughout the majority of the season, the reservoir was mesotrophic. And then in all three basins, you can see that it kind of goes eutrophic towards the end of the summer, which is to be expected. But the key takeaway from these plots is that historical Park Service data hasn't shown oligotrophic values in Savonniere and Saboya. So that's like a huge thing that we want to look at in the future. And these graphs I'm working to put into a more digestible figure currently, but there are preliminary zooplankton data. The general trend with these ones is that you see a higher zooplankton abundance in that photic zone, and then as you move down, they like decrease. And then, so because I'm not able to share our phytoplankton identification yet, I have taken a few pictures of our cyanobacteria species that we've seen. These are kind of our three main offenders. We have Dolichospermum, which I think is the prettiest. It kind of looks like a pearl chain. And then we have Lochia, which are like little puffballs. And then the Aphanizomenon historically has been our number one offender. And you can really see those, I don't know how to use the pointer, but the vegetative cells are the sites of nitrogen fixation, and that's the big problem. And then this is our vertical gill netting data. The biggest takeaway from this one is that you can see in Saponiero in the pelagic zone, it's mostly lake trout because that's where they like to hang out down in the depths of the reservoir. And because we're seeing those kokanee move to the bottom more because of the zooplankton abundance, they're kind of increasing that predator-prey squeeze down at the bottom. So you're seeing more predation on kokanee from lake trout. So looking to the future, we really think it's important to do continued monitoring and research. This past year that we did research was not a very high algal year. We didn't see many toxins. It's kind of more of a baseline view of the reservoir. So I think it's really important as new technology comes out to keep monitoring the reservoir for consistency so we can get a long-term data set looking at mitigating the impacts of the algal blooms.
Thank you so much, Lucca. One person from the audience wants to know if you have a favorite phytoplankton. I know those are still in the works, but do you have any preliminary ideas?
I do, actually. There's a really pretty diatom called Fragillaria that Martin and I have been seeing in a lot of our samples. I don't know how to describe it. I wish I had a picture. It's just kind of like that.
You can send it to us later. Do you have any hypotheses about why you saw those oligotrophic values?
Yeah, that's a great question. So that study that I mentioned by CPW that's been going on, in the past decade, they've been seeing actually negative DO values at the bottom of Ceboya, and that's resulting in a lot of the fish kills that we've been seeing. So we don't know why, but we have a lot of data that shows that Ceboya is very oligotrophic.
Is there any relationship between algal blooms and seasonal snowpack?
That's a great question as well. It definitely depends because the snowmelt has a big impact on like the amount of water that we see in the rivers and the reservoirs. So with increasing water, you can see more dilution of those toxins. So I think when we have higher snow years, it's harder to do algal bloom research.
Kind of leading off of that, what causes some years to have larger blooms than others?
Yeah, so those years are real low water years. So I think it was like 3 years ago in Blue Mesa that you could go out on any day and it would just look like green sludge. And that was pretty much just because the water year was so low that year that the sunlight is really able to penetrate deeper and get more of those species.
So from what you've learned so far, what do you think the most important next research steps are?
I think definitely looking at the water tables and finding a year where we're predicted having a low year, because getting that data where we have all of that high concentration of cyanobacteria is super important for understanding as well.
Awesome. Any final questions from the audience? Pat, shout it out. I'll repeat it.
I think I could be loud enough. What does your research tell fishermen about fishing? Who makes the
Yeah.
dangers or impacts of the
Yeah, the importance of this research for people who fish.
I think that vertical gill netting data is going to be super important in telling that maybe we should be looking at permitting more for fishing. I know Blue Mesa does a huge tournament, like a fishing tournament on the lake every year. And I think they're looking at altering that based off some of our data, but we don't have enough conclusive data to really say anything. It's more of like an observational study.
Amazing. Well, thank you so much. Big round of applause for Lucca. All right, our last presenter before the group panel is Megan Locher.
Thanks, Susan. Okay, awesome. All right, I think we've already talked about the importance of wetlands and restoring them, so I'm not really going to belabor that point. So I'll just kind of dive right into talking about monitoring the impacts of wetland restoration efforts in the historic floodplain. And I'd like to kick us off with my acknowledgements, namely Madeline for being an amazing mentor, for Phil for being super patient in my learning about aerial photography. Tim, thanks for helping with all the coding. Julie for being amazing in teaching me how to frame my project and maintaining the scope of it. And for the Save the Frogs organization for a grant that helped out with the amphibian section of this, and for the employees Clark School program that we're all in. So yeah, before I jump into details, because I tend to do that, I'd like to talk about the overview. So Cold Harbor, my study site, is 7-8 miles east of town. And you can kind of see the US Highway 50 going across this. That works. Nope. Going across that way right there. And I focus my study mostly in this phase 1 area, so outlined in blue over here. And zooming in on that location, that's weird. I just want to highlight that, so I'm talking about restoring a wetland area, and that implies that something has happened to it, right? So I wanted to highlight the 50 again, going across
east to west, I guess that point was dead. And there's a historic railroad grade that goes right here where that yellow line is, which has kind of created like a berm in both locations, preventing like a nice flow of water, sheet flow across this area, right in those locations. And to try and kind of fix that, I should say that this project is at least 5 years old. And so I'm standing on a lot of other people's work, which is fantastic. And to try and like ameliorate these impacts of the preventative sheet flow in this area. In 2023, the student that had this project before me, with the help of a ton of volunteers, installed BDAs in these locations. And we've already learned what BDAs are, but they're just a low-tech process-based restoration technique, just trying to slow water as it moves through, in our case, this irrigation ditch. You can kind of see right next to all my pink stuff right here. But basically you just take some vertical posts, pound them into the ground, weave in some willows in between that you harvest or whatever else you have natural material wise around. And again, another map of the location. I'm going to use this red box a little bit later when I talk about the aerial photography section. But yeah, just a really fun way to look at the landscape, I think. Wow, the formatting got weird. So I think of this project in 4 different sections. So there's soil moisture, aerial photography, vegetation survey, and amphibian surveys. So I'll go in that order. And starting with the soil moisture data, I just wanted to focus on these these blue points in that same area that I just showed you. That's where I took all of the soil moisture data. And again, another map showing the numbers that associate with those points. And then the average values that I took from all of those locations. So on the left, you just have the table of at each of those numbered locations, the average value across the season for 2023 on your left and 2024 on your right. And then up top, that's 2023, 2024 on the bottom. And that's the average value for each week across the season. So you can kind of see that it held on to a little bit more moisture in 23, and then it dropped and it had some spikes, which I think has a lot to do with like snow melt timing, sorry. And the flood irrigation from the neighboring ranchers, because you know they'll flood irrigate to help with ranching, and then that water moves across into Cold Harbor. And same thing with 24. It was just like a longer monitoring season, so there's more points. And another way to look at that same data is just seeing the differences. So anything in that dark purple kind of black color indicates an increase from 23 to 24 in average moisture levels. Anything in yellow indicates a like loss in average value. And then moving on to the next section, the aerial photography. I think these are like the coolest maps ever. So there's 2 columns right there, 23, 24. You can see the difference in the 2 months. So we took photos in May, July, and September each year. And you can kind of see there's not like a huge difference in like May, they kind of look the same, as opposed to September in both years. You can see it kind of looks a lot less green in 24. So I thought that was kind of interesting. But it could also have to do with like the weather that day, or there were more clouds. But then from there, in this pretty dark graph, sorry about that, we were able to statistically find if there was any differences in the greenness. So remember that red box I showed you a few slides ago. So looking at values just within that box, we were able to see that there's a very small but significant increase in greenness in both May and July, and a slight decrease in September. And moving on to the third section, the veg surveys. This is a very, very blown up map. I'm sorry, it's very blurry, but I just wanted to highlight the exact locations of where I did these surveys. So basically what I did was take this square PVC 1 by 1 meter square and just plopped it at the coordinates, and then identified every plant within that square. So the results that I'll talk about are based on diversity, not on percent coverage, which I think is a lot more common, but I just want to highlight that change. And from there, man, this was a feat for us to do. But at each coordinate, we were able to figure out kind of what each plant within those plots preferred what type of ecosystem. So anything in orange prefers drier soils, green generalist, so wet or dry. And then anything in that blue-black color is prefers wetland. So this is hard to kind of talk about because it feels like it's all over the place, right? But I do think it's also important to highlight that while our irrigation ditch runs this way, you can kind of see it's a little bit green there too. So just ecology, nothing is perfect.
So I think it's really interesting preliminary stuff that we can just take a look at. And again, a different way to look at this data, anything in orange, dry, green, generalist, blue, wetland. You can kind of see that on the very, very left here, this is all based on distance to the irrigation ditch. So the one on this very left is the one that's geographically closest to the irrigation ditch. The one on the right here is the one that's geographically furthest away from the irrigation ditch. And again, ecology, nothing's perfect. Water goes where it wants. So it's really hard to draw conclusions, but I think this is really interesting. You can kind of see that the wetland plants are leaning towards the ones that are closer, but that could just be me. And then moving on to the last section, amphibian surveys, of course. So this is just a heat map of the northern leopard frogs that I saw all across the property. They were mostly right around here, with a couple of other individuals found at a later date, not during amphibian surveys. And then boreal forest frogs, which are the ones you're going to be hearing in about 2 weeks, and I'm very excited for. And mostly they were found kind of in that same area, but also a lot more found kind of towards the eastern side of the property. And I had originally planned to do 3 times as many surveys as I was able to do, but there's very scary moose. So I was scared off and didn't want to risk a broken collarbone. So I applied for a grant through Save the Frogs that I talked about earlier, and actually got it. So it was really exciting. And I was able to make a rack card and a sticker. Give me like 2 weeks, they'll be out. I know everyone really likes stickers. Just to try and like help with the enthusiasm with amphibians that I have with the Gunnison community. And so that, yeah, like that'll be in a couple weeks. And then next steps for this project, I think everything that Laura highlighted with BDAs and this type of restoration work is that it takes time. It's been a year. So we need more time to see any more definitive changes to see, are these working? You know, are they failing? What's going on? But yeah, I think this was really interesting to look at just after 1 year. So thank you.
All right, thank you so much, Megan. One question for you. It looks like your study sites are kind of in that corner between 50 and 114. Do you think the roads have any impact on the rate of change?
Rate of change.
Like restoration or?
Oh, interesting. Do the highways have an impact on how well the BDAs are working? Is that kind of the question?
Sure, how?
Interesting. Yeah. That's an interesting question because we do have a lot of, this is just insider knowledge, but there's a lot of willows right next to those highways. It could be because of the highways, and willows are pretty hardy and they do really like those wet areas. And there's another irrigation ditch that goes north to south right next to the 114. So potentially, right? Because if the willows are right there, they're eating up all the water, they're preventing even more water from being in the irrigation ditches. Potentially, right? Because the more water you have, not too much that you're destroying BDAs, but the more water you have, the more chances you get for it to hit the BDAs and for the sediment to drop and kind of build up the BDAs, which is the point of them.
Thanks for the insider knowledge. How do you conduct amphibian surveys?
Oh man. Yeah, so I had this really great plan. I checked out a book from the library and everything. I had these really nice square like survey lines I was gonna walk. And then I got there, it was like 11 or 12. I get there, I have my waders, I'm really excited. I step in the water. I got stuck. So those went out the window really, really fast. So what I ended up doing was just walking along where there's water. And so I broke up, I didn't include this in the presentation because of time, but I broke up the whole property into 8 different sections and was going to survey each of those 8 sections 3 times. But yeah, the moose cut that short.
Gotta take care of yourself there. So what about the frog species you found? Were they in the places that you expected to find them?
Yeah, I think I expected to find more in the irrigation ditch areas, but maybe the water was moving a little bit too quickly for them. With the chorus frogs, that population kind of on the right side that I highlighted, I called those the Finger Lakes because they just kind of look like Michigan, you know, like that. So there was a ton in there, and I didn't even see that coming. So quite a surprise. It was nice surprise.
What's your favorite type of frog or toad?
They're all jealous because I got the really good question. I really like glass frogs. They don't live in the US, but if they're like stuck on like, if you have them as a pet, say, and they're stuck on the glass, you can see all their organs and their heart pumping. So they're adorable.
What management recommendations might you derive from your findings?
Right, yeah. So BDAs are pretty go-to, I think, right now, as well as like low-tech process-based restoration in general. I think BDAs are just a really kind of famous version. They're really nice to look at and everything. So in terms of management, I feel like what I just highlighted at the end and what Laura highlighted is just to continue monitoring, and that when you apply for a grant or when someone is overseeing a project, to not expect results right now. This is a long-term process based, like it takes time. It's a naturally based restoration. You're not going out there and fixing that today. You're fixing that over the course of the next 5-10 years.
What about water year? Would you recommend repeating these studies across different hydrographs?
Just patterns of water over different years. I'm not a hydrologist. So, and I think we talked about this earlier, how certain years, especially in Gunnison and a lot of other places, I assume, we have really wet years and really dry years, and those influence our ecosystems pretty heavily. So, and then there's the timing of the season as well, right? Which is highlighted in the graphs that I showed you with the soil moisture when you're talking about the whole season, the average values and how that changes over the season. So I think it's good to be aware of that when you're thinking about results, and that was highlighted in the aerial photography as well, how like May was really kind of brown and gray, and then September was really green. So I think it's good to be aware of that kind of thing. There's not much you can do about it, but again, with like the long-term planning, just be aware of that.
Awesome. Thank you so much, Megan. Round of applause. All right, what a fantastic panel. So now we have time for questions that we can address to the whole group, or continued questions for 1 or 2 folks at a time. So feel free to pop those into the QR code. I'll move over here so that we can just pass the mic. So what are your post-grad or post-thesis plans?
Sure. Yeah, so I'm very excited to be working with Full Harbor as the program coordinator. And I can't express how excited I am to do that, because I love this work and just feels like a natural upward progression. So thank you.
As some people might know, I got really involved with theater here in the Valley, and I scored a full ride to acting school. So that's where I'm headed, in Savannah, Georgia.
Do you want to plug your show tonight?
Oh, yeah. So there's a show, it's called Clever Little Lies. It's a comedy about a dysfunctional family. And it's tonight, tomorrow, and Saturday at 7 p.m. at the Gunnison Art Center. Then next week, there's Friday, Saturday, there's 2 improv shows in Crescent Butte at the Mountain Theater. And I'm also directing my first show at the Mountain Theater, which is premiering at the end of May. And it involves a ladybug in one scene. And then also in June, there's a couple clown shows in Crescent Butte. And in July, I'm going to be in Last of the Red Hot Lovers by Neil Simon, also in CB.
I'm planning to continue working for the Park Service at Cure County, doing water quality work.
I am also I'm working for CPW, but with the Park Service with Lucca. And I plan to do that for at least most likely 3 more seasons. But yeah, I would love to go anywhere and do some sort of water resource management.
Awesome. Congrats to you all on all of your new plans. What recommendations do you have for future students interested in conducting similar research?
Advice to leave that? Yeah. Just trying to think. I guess I could just tell my own story. Moving into grad school, you know, you have a few months to plan and everything. Just trying to figure out what projects I was going to do, honestly, for both. And I feel like I really lucked out in being able to study things that I was really excited about and gain some skills that I didn't know I needed. So my advice would be to just ask, because no one here is a herpetologist yet. But we still managed to make it work, and I got to study some really amazing things. So always ask. The answer can only be no, right? Or yes. And you can end up doing stuff your dreams, you know.
For me, it was a little different. I had never worked with bugs before, and so I had no idea how to go about that. So I would recommend be open to anything, because now I'm a bug person, and also try to familiarize yourself with what you're studying before you're actually getting into your own samples. So I got to practice a lot on identifying bugs, which was really helpful.
Yeah, I would say just be open-minded and flexible. A lot of logistics with my project kind of got switched around the whole time, and you just kind of gotta adapt and make sure that you're really focused on the importance of what you're studying.
Yeah, I would just recommend reaching out to many people at the places you're considering going. They're obviously going to have all that insight that you want, and you'll get an idea your research is even possible there. But yeah, like they said, just being open-minded to anything, and you might become really interested in something that you never thought you would be.
All right. How do you think your studies work together to inform management decisions? You can take a minute to think about it.
I think that because freshwater is our most crucial resource, and all of our projects might seem vastly different, but they are all connected to kind of inform of the degradation of freshwater and kind of educating the public about how important it is to preserve those ecosystems.
I think at least like Megan, Laura, and my project, we all have a restoration component to our projects, and that is so important for wetland habitats. But just like Lucas said, studying them in general, just to know what changes we're seeing.
Yeah, maybe Lucas will help us restore algal bloom systems. You never know. We'll pull you into restoration. All right, so all of these study systems are degraded in some way with various efforts to restore them. What are the longer term outlooks for each system given various pressures? What do you think is going to happen in the future of each of your systems?
I feel like the giant elephant in the room is climate change right now, right? All of our systems are going to continue changing. And I think maintaining an adaptive plan for going forward with these restoration projects is going to be really key, because we can predict and map all we want. I mean, I'm one to say that my thesis is predicting and mapping, but we can only plan and map so much versus being out there continuing to monitor and make changes in our planning based on that. And I mean, we do plan to continue restoring in different areas in those different phases that I talked about. Yeah.
I think specifically for my study side, it's really high up in elevation, so it's very vulnerable to climate change. But I think that the BDAs has been a huge effort, because it's over 200 structures that have been installed. So there is immense effort and budget going in there. So I'm really hoping that they continue that, and I think that they will. And I hope there's going to be more research to ensure that these projects are going in the right direction. And as we've seen in those pictures, that physical characteristics have already changed in those couple of years that have passed since BDA installation. So I'm optimistic that it will continue to go into a positive direction.
Yeah, I think talking about Blue Mesa in general, it's a multi-agency managed reservoir. So we have a bunch of different government agencies really focused on the health of that ecosystem. I do think, like Megan says, as climate change continues to increase, we'll start to see more frequency of these blooms. But I think we have so many hands-on with this one reservoir. It's kind of being like the prize child almost.
Yeah, as Megan mentioned and I mentioned in my presentation, climate change is so critical for my fens because as soon as that groundwater is depleted, all of that process is reversed. So that's obviously concerning, but aside from climate change for my project, we're hoping that those mine adjacent sites go back more to their historical conditions and we start seeing a healthier macroinvertebrate community move into that site and start matching the reference site. But of course, yeah, climate change is a big one.
All right, one last question to close out the panel. So I often found during my graduate research that the places that I studied became really personally important to me. So I'd just love to know your thoughts on, how you feel about the places you worked and how that feeling will inform places that you go from here.
Yeah, I mean, whenever someone says the word wetland or riparian, I get a little excited now. And yeah, Croll River is very special to me. And I found it so, I don't know, fulfilling to continue to work out there and to like see the changes. We had like a beaver move into one of the BDAs, so that was really cool. And like just a regular dam. So yeah, it does definitely become special to you. And I think that has changed the way, like the forward motion that I have and what I want to do career-wise, like in the next, 5-10 years.
And for me, it's really changed the way I look at wetlands and now knowing all the different important factors the wetlands have on the general ecosystem. And I lived in New Orleans before. There's a big crisis with the wetlands there, which is very different than here, but there is connecting facets of it. So it's really interesting. And I'll be looking at it in a different way from here on. And yeah, I've enjoyed working with bugs and I'm a bug person now and wetlands. And I hope I can come back to Trail Creek this summer to help with sampling that's going to happen there this summer.
Yeah, I'm from the East Coast, full disclosure. So I moved out here for the job that I have and for this degree. So I spent 5 years on Blue Mesa, just kind of boating around and sampling, and it's become kind of like a second home to me. So much so that I don't know if I'm able to leave Blue Mesa. So yeah, it's just pretty cool place.
Yeah, I had never done any work with fens before and I'm really enjoying it. It's a little bit of a jaunt to my site, which makes it even more exciting to go because not everybody gets to see locations like that. Yeah, it's just really lovely to be in such a beautiful place. And also the people I've met, like everyone at Mountain Studies Institute, it's been a really great experience and an opportunity to make really great connections.
Awesome. Thank you all so much. This was a great panel. Another round of applause for our Freshwater Women in STEM. Amazing. All right, so we are headed into a break now and we'll